Great. So thank you very much, Manjunath, for your time today. It's great to speak to you today about your work and also our upcoming event. Please, can you describe your role at GlaxoSmithKline?
I'm a scientific investigator and a GSK Associate Fellow. I'm a part of a department called Bioform Discovery, which basically discovers and optimizes antibodies against different therapeutic targets. So as a scientific investigator, my role is to provide technical leadership in 3 core areas. One is evaluation and technology landscape analysis, which means looking at the technology that's out there in the external environment, find the one that fit our overall strategic priorities, and evaluate them either through formal collaboration or small proof of concept studies. Second aspect is implementation of new technologies. Once we bring in a new technology that's promising, we still need to make it more robust and fit for purpose for our needs before we can completely embed them into our discovery workflows. And the third aspect, which I'm truly passionate about, is developing new tools and technologies for drug discovery, especially now for single-cell research. And also as an elected fellow in GSK, the expectation from my role is to be a bit more science-focused compared to non-fellow scientists within the organization.
Great, thank you very much. This sounds very interesting. How did you get involved in single-cell analysis, and why did you decide to look at this specific area?
I have a background in engineering and cell and molecular biology. My expertise lies in using microfluidics or microtechnologies for drug discovery. And since most of single-cell research involves use of microfluidics-based tools, so it was natural for me to get into this area. So when I started my scientist role at GSK, my job was to develop nanowell-based microfluidic tools for single-cell analysis, mainly to look at immune cell interaction in the tumor microenvironment, which involved looking at heterogeneity of single primary immune cells, both at functional and transcriptomic level. The role has now changed to focus mainly on single B-cell analysis, both at phenotypic and genotypic level, purely for antibody discovery applications.
Thank you very much. And what are your current priorities at the moment?
I'm currently focused on exploring high-throughput single-cell tools and technologies for both screening and NGS-related applications for antibody discovery against a variety of different targets. So mainly I'm looking at high-throughput single-cell analysis tools.
And you are also heavily involved in looking at single-cell analysis tools for genomic transcriptomics and transcriptomic and proteomic purposes. Could you please tell us a little bit more about your work and recent findings?
Sure. When discovering lead therapeutic antibodies involves 2 different aspects, and we need the best cutting-edge tools for both of this. One is screening, where we need to profile a large number of single cells, either primary B cells, hybridomas, or if you take in vitro display formats like yeast or phage that are expressing a range of different types of antibodies. And you want to find those rare cells that are producing target-specific antibodies. It's like sorting like a needle in a haystack. So the second aspect is sequencing, where we need the sequence information of antigen-specific antibodies, or we want to understand the antibody or B-cell repertoire, say from an immunized animal or human, by looking at the whole transcriptome or B-cell receptors. So there's 2 aspects, screening and sequencing, and looking into both aspects of it and using single-cell analysis tools for that.
Thank you. What opportunities do you think the future holds for single-cell research and tools development, specifically for the above areas? How do you think the field will evolve in the future?
The future looks very promising. I mean, there are more and more tools being developed for mining single cells at genome, epigenome, transcriptome, metabolome, or even phenotypic level, and to get deeper insights into disease biology. I think most of these screening and sequencing sample preparation tools are relying on sophisticated fluid handling and manipulation techniques based on microfluidics. There's also a new suite of tools being developed for sequence data analysis at a large scale. So many of these are getting commercialized too. Hence, the entry barrier for single cell researchers has been lowered. So you don't need, for most biology lab, you don't need specialist microfluidics engineers or core bioinformaticians to use any of these tools successfully. So I see a lot of positive developments and a lot of cutting edge technologies on the horizon.
Thank you. And in your opinion, what are some of the major challenges that pharmaceutical companies are trying to overcome in studying single cell analysis?
I mean, taking challenges in general, I think single cell technology landscape, like many other technology landscape, is evolving so rapidly. Many of these technologies are not entirely validated before it's launched into the real world. So since many of these are not very robust or fit for purpose for industrial applications, we as end users end up spending a lot of time optimizing it, making it more robust, which is a challenge many of the time. So my advice to a lot of developers or academic entrepreneurs is to engage with their stakeholders very early, try to understand the use cases and case scenarios very early before they go on to develop instruments or technologies. I mean, talking about specific challenges we face, I think when you perform omic analysis to get deeper and clear insights into what's happening within a population of cells, for us, the challenge is finding specific therapeutic targets based on that information that are druggable, targets against which we can develop medicines and treatments. Whether we go analyze single cell data from published literature or from non-profit or for-profit sequence databases, we need to be able to find unique targets for any disease we are trying to treat, and that should be commercially viable too. The second challenge is for antibody discovery in general, we often need to screen millions of cells to find rare cells that are producing antibodies of interest that meet the quality criteria of what makes a good medicinal therapeutic antibody. It needs to have certain affinity, it needs to have certain functional properties. So it's like finding a needle in a haystack again, as I said. And that needs higher throughput screening and sequencing tools than the ones available commercially currently. So we do not have the freedom to go ahead and get in technologies or protocols or tools that are still in development or in academic literature. We just don't have the freedom to operate. So that adds an additional challenge for us as a pharmaceutical company.
Thank you very much. And what would you like to achieve at our Single Cell Analysis Congress this December?
I hope to learn about exciting next generation single cell tools that are on the horizon, the unique focus of this Congress in general on single cell research and NGS aligns very well with my current research priorities. So yeah, I'm just hoping to learn a lot.